first of all, let's move on without further ado and go to look at our first film, which is a bit of a thriller, really. It involves an invisible enemy that's all around us. So the question is, can you guess what that enemy is before our scientists reveal what they're working on? Thank you.
Since the dawn of humanity, plants have been vital to our survival. We consume plants directly as grains, pulses, fruits, and vegetables. And we rely on plants to feed our livestock to produce meat and dairy. Every year, we cultivate billions of tons of plants for food, materials, medicines, and fuel. With the population predicted to rise to at least 9 billion, it is estimated that we will consume twice as much food by 2050, while our food is under threat by an invisible enemy.
From above ground, these potato plants look okay. They occasionally show signs of dehydration, but these plants are well watered. So how can this be?
At first look, there isn't anything visibly wrong. Maybe we need to dig a little deeper. The roots of this potato have been heavily infested by a mysterious pest. A pest that is stealing the plant's nutrients and controlling its development. To understand what is happening, we need to go into the lab.
Here at the Crop Science Centre in Cambridge, we study how peculiar pests
make the plants sick and develop methods of controlling them in a sustainable way. Here is a plant parasitic nematode, a microscopic worm capable of infecting crops and causing devastating plant diseases. Nematode worms are some of the most diverse and most abundant animals on Earth. In fact, for each human, it is estimated that there are around 60 billion nematodes. If we were to take away all the matter on Earth and left only nematodes, we would still see the outline of the entire planet. While most nematodes are an essential part of the ecosystem, they also infect our most important crops. For example, take this burger here. The bun comes from wheat. There's a nematode that infects this plant. The beef is produced by feeding cattle with soybeans. There's also a nematode that infects this plant. The lettuce, the ketchup, the onions, even the fries, they're all infected by at least one nematode. In our lab, we study the Cyst Nematode, which infects plants including tomatoes, potatoes, and soybeans. Unlike most animals, the sex of a juvenile nematode is not determined at birth. So how does it work? It's decided by their success. Those that are most successful in getting nutrients from the plant will become females, while the others become males.
If we can understand what makes nematodes successful, we can find ways to control them. In this way, we hope to find a sustainable way to control this tiny but powerful parasite and help feed the Earth's population for years to come.
Nematodes use several weapons in forms of protein in order to acquire nutrients from the plant. But the plant is not defenseless. They possess their own set of proteins to fight back. It's like a battlefield. The nematode and the plant are in an arms race to fight each other off. One of the major goals of the research in our lab is to ensure that the plants will have an upper hand in this fight. We try to understand the weapons nematodes use to invade the plant, how they control these weapons in time and space, and how we can harness this knowledge to teach plants how to better defend themselves from nematode attack. Our future is bound to and is bounded by plants. So the next time you are at your dinner table, give another thought to the hidden enemies that threaten our plants.
Okay, well, that was absolutely fascinating. And I'm amazing to sort of to realize the sheer numbers of nematodes and the problems they can cause. It'll cause us all to look again at the plants growing in both in our gardens and the crops growing in the fields. I'd now like to introduce the team that are behind this research. There's Dr. Sebastian Yves van den Akker, who's head of the Plant Parasite Pathogen Interactions Group. And he's got some members of his team with him here tonight. There's Dr. Inati Sunavala, and have we got Olaf as well? No, oh, Clement Peregrine. Welcome, Clement. Thank you. Okay, so do we have any questions? Have anybody put any questions into the Q&A session? I haven't got any at the moment. I've got a question. So how you mentioned that we need the sex of the nematodes is determined by the amount that they feed. Where exactly do they feed, and what are they taking from the plant?
Sure, I can answer this question. I mean, most plant parasite nematodes feed from the roots, which is why we kind of call them this hidden enemy, because you typically don't think about them. And they'll feed from the center of the root. And one of the things that we find that's quite remarkable about what they do is they transform the root into this novel kind of organ that grows up. And the way I like to describe this, it's the wrong word, but it's got the right connotations. It's like the worm can make the plant make a tumor, and then it slowly feeds on the tumor. Which kind of highlights this really destructive power of these organisms to manipulate plant biology and also kind of immunity. So that's what they feed from plants, and they kind of direct both plant water, but also the plant photosynthate, so the sugars that the plant can make from photosynthesizing, and they'll withdraw all of this through that single cell into the nematode. And as Unati was saying in the film, if they're really successful in this, then they'll develop into a female, and this kind of makes sense because females have a lot of high energy requirements in order to produce lots of eggs, and if they're kind of unsuccessful, then they'll develop into males because they simply can't support that kind of growth.
Okay, thanks. There has a question. So then, just to confirm, then that we've had a question that's come in about that. Just to confirm that the nematodes are then born sexless, as it were, and so how does that determination occur? You mentioned in terms of just nutrient availability, but there presumably must be some sort of signaling process that's going on.
Yeah, we have the same question, so we don't know. So yes, they are born sexless. They are born juvenile, and so they don't have sex chromosomes or anything like that. And we don't know how they know. We don't know what the signaling events are. This kind of environmental sex determination is pretty rare in general in life, in biology. But to have it linked to food availability is even rarer. So you may have heard of some examples of environmental sex determination. For example, if some reptiles, if their eggs are incubated at a particular temperature, then they'll grow into preferentially females or males. But to have it linked to food availability is super, super rare, and we simply don't know how it works. And yet, if we did know, it would be important. Because if we could manipulate this sex determination and make sure that the population is entirely female or entirely male, then we wouldn't get another generation. And so it's a super interesting but also super important question.
Okay, so there's some questions coming in now, which is great to see. So can nematodes ever be beneficial? One of them is asking. Who'd like to take that?
I was going to give the other guys a chance to speak, but I'm happy to take it. So, I mean, yeah, most nematodes are beneficial, the vast majority, massively so. However, most of the nematodes that are studied are the parasitic nematodes, either of plants or of animals, because of the huge burden that they incur on either food security or on global health. So, yeah, I think most nematodes are beneficial, and indeed they can be really useful indicators of soil health or environmental health. So the kind of complexity of the lifestyles of nematodes can reflect, how healthy a particular field or a particular, area of forest, for example, can be.
Okay, so maybe we could ask Inati or Clement. There's a question here as to whether why you're focusing on nematodes rather than some other plant pathogens.
I feel like this is the kind of question that Sebastian should answer, really. Why are we focusing on nematodes? Well, they are an important plant pathogen and somehow underexplored. Most probably because, you know, they are sort of hidden, like we're saying, and you don't really see the symptoms until much later. Whereas, you know, let's say if you have bacterial diseases on leaves, let's say, you would see them much faster. Important pathogens requiring understanding, and we're working on it.
Yeah, same thing. Yeah, I think I totally agree with Funati. Basically, they may look a bit like neglected compared to other pathogens, like fungi, for instance, or mycetes, but actually they have a very significant impact on the economy. And so because of that, they very, really deserve some heavy research on them. And so it's particularly interesting to work on them because there are so much things we don't know about them. So I think this is what actually drives us. Yeah.
Brilliant. Okay. And one final question we'll take those. You mentioned, it's just come through, how might you mentioned how the research is being used in your lab to fight those pathogens. And so apart from this sex determination issue, the question asks, what are these attributes that you mentioned? Plant defenses, how do they come into it and how can you enhance those?
I can answer this one, I guess. So, I mean, we're basically trying to understand any area of this plant parasite interaction in enough detail to identify routes to control. That's the kind of the motto of the lab. Everything we do is revolved around that. And for an obligate pathogen, so an organism that can only be a pathogen, that means that pretty much every aspect of its biology can in some way be used to identify route to control. So to give you an example, you know, there are some genes in the plant that the nematode needs to be present in order to cause disease. And this sounds like a weird concept. This is the idea of a susceptibility gene. So the parasite will need a particular gene to be present inside the host in order to cause disease. And so, for example, if we could remove that gene from the plant, we could render it immune again. And so, you know, we've got, that's just one example of one of the ways we could use our fundamental understanding to deliver, you know, an applied outcome. Brilliant. Okay, well, thank you so much to your team. Thank you, Inati. Thank you, Clement, and thank you, Sebastian. And if I may, just before we close, I have to say thank you to the other people that couldn't actually be here today, but were involved in the film, particularly Olaf Krantz, because he did a lot of the work on this. Okay, brilliant. Thank you, Sebastian. Yeah, and just to say that there are some questions that we didn't get around to answering, particularly whether nematodes are a threat to humans or people's gardens. Perhaps you could pick up on some of those in the chat function. That would be brilliant. Okay, well, thank you very much, Sebastian and his team. Now our next film involves a seemingly unremarkable structure, which you may be able to find down at the Botanic Gardens here in Cambridge. But what secrets does it hold? Let's go and find out.
Hello, my name's Hamish, and I'm a PhD student in Professor Beverly Glover's lab in the Department of Plant Sciences here at the University of Cambridge. Today, I'm going to be showing you some of my research, which is using the research area here at Cambridge University Botanic Garden. This is somewhere that the scientists are usually allowed to go and play with experiments, but the general public are kept out. So let's go and have a look at what we've got in there now. So I'm working inside this polytunnel, but before we go in and have a look, I want to tell you a bit about why I'm doing this research. About a third of the food that we eat depends on insect pollination to some extent. And insect pollination is where an animal like a bee or a fly visits a flower, collects pollen and takes it to another flower to fertilize it and help it grow food for us to eat. Now, as the world population grows, we're going to need to find more food. So we're going to need more insect pollination. But around the world, insects are generally in decline, and those two things don't really square up. We're going to need more insects for pollination, but there aren't so many of them around. So while zoologists and ecologists are working on ways to reverse this insect decline, I'm looking at the other side of things. How can we make plants more efficient for insects to find and more rewarding when they get there? And this experiment which I'm doing here is looking at the more rewarding side of things. I'm trying to find out how much nectar and pollen different varieties of strawberry make And then that will let us know whether we can breed for varieties which make more nectar or more pollen. But first of all, we need to know this variation that exists naturally. So have a bit of a think, how many insects you might be expecting to find inside this polytunnel where I'm doing my experiment. Have a think about that, then we'll go inside and have a look. Over that. Yeah. So that was actually a bit of a trick question. Now we're inside the polytunnel. You can possibly see or hear there aren't any insects in here at all. And that's because I've got insect-proof netting all the way around the sides and all over the doors as well. I don't want any insects in here at all, because what I want to find out is how much nectar and pollen these strawberry plants can make when there's, and the only way to do that is to measure it. And if there's insects inside who are eating it, then I'm not going to find out how much it is, because the insects will have come and taken it away already. This is a problem I discovered in the field last year. So I've put up this polytunnel here in the Botanic Garden to be completely insect proof, and I can grow my plants in here and then measure the nectar and pollen that they produce. To do this, on the first day that I'm doing the experiment, I will mark the I will pick all of the flowers which are currently open, because I don't know how long they've been open. On the second day, I will then mark all the flowers which are now open, so I know that they've been open for a maximum of 24 hours. And then on the third day, I'll pick those plants which I've labelled, and then I'll take them back here to my microscope, and I will measure what's going on in there. To do that, I pick all of the stamens from the plant. The stamens are the bit of the flower where the pollen is made. Put them in a tube, and I stain them with a purple stain. And then back in the lab, I can count how many pollen grains there are. And because, yeah, that will tell me how many pollen grains there are for flower. But also what I do while I'm here, once I've picked the stamens, is use a tiny, tiny glass tube, which holds 0.5 millimeter cubed. That's a really, really tiny amount. And I use that tiny glass tube to suck out the nectar from the flower. I have to do that under a microscope because they don't produce very much nectar, and it's in a slightly inaccessible place. And then when I've done that, I can measure how much nectar the plant produced, and also I can measure how sugary it is. And using those lets me calculate how much energy the plant is making. Because obviously if one plant makes one quantity of nectar which has 20% sugar in it, if another plant makes the same quantity of nectar but it's got 40% sugar in, that plant is twice as good for the bees in terms of the energy they get to bring back to the nest. So once I've measured all of those, I can see if different varieties of strawberry produce different amounts of nectar or pollen. I've got 20 different varieties of strawberry growing in here at the moment. Sadly, I'm still in the middle of the experiment, so I can't tell you what the result of that is, but hopefully at some point in the near future I'll be able to. So now I know how much nectar and how much pollen these different plants can produce. We need to see if it's useful to the insects. And to do that, we need to go back to the Department of Plant Sciences. So I'll see you back there. So I'm now in the Department of Plant Sciences here at the University of Cambridge, and I'm going to take you into the lab behind me to introduce you to some of my friends who help me with these experiments. Let's go and have a look. So here we are inside. It's quite a small room here, and you'll see next to me I've got this little green box. Who do you think might be in there? Let's have a look. So this little tube here is the entrance or exit for a colony of bumblebees, which we keep in this cardboard box. We buy them from a company in Ashton Keynes. They arrive in a small buzzing cardboard box in the post. And we use these bumblebees to help us with these experiments and tell us what it is that they think. So I'm going to talk to you today about the Nectar Experiments, which I've done, and also which somebody else, a previous PhD student in this lab called Emily Bales, has done. Now, bumblebees can learn to associate a visual cue, something they can see, with the quality of the reward. And to demonstrate this, I'm going to first of all get you to have a go at something. So here you can see I've got a setup where I've got 6 different coloured discs, and 3 of them are yellow and 3 of them are white. And I want you to choose one of the colours. Just choose one of the discs. And the aim is to try and get a sweet and tasty reward. You chosen them? Chosen one disc? Excellent. Okay, I'm going to now show you the reward. Well done to those of you who chose the yellow ones. Okay, let's try it again. I'll cover them all up again. Pick a colour and any colour you want. Okay, ready? I'm going to show you the rewards. Again, well done to you who chose the yellow ones. Okay, I'm going to cover them up again. Pick a disc to try to get the sugary reward. And I'll uncover them. Well done again to you who chose the yellow ones. So you'll see we're getting a pattern here that you're learning to associate the yellow discs with the sweet and sugary reward, and learning to ignore the white discs which have the reward you don't want. And it's the same with bumblebees. I can use this little card which I've got here, which has one white circle and one yellow circle printed on it. And I can put some sugary water on both of them. And on one of them, I'll put a sugary water which has more sugar in it. And on the other one, I'll put sugary water which has less in it. And because bumblebees can learn to visually associate the color of the disc with the reward that they get, if they can tell the difference, and if they prefer the more sugary reward, then they'll go to the yellow disc where I've put the more sugary reward. If they can't tell the difference, they'll go half and half to each of them. And actually, this is what we find. We find that the bumblebees do choose a more sugary reward over a less sugary reward, all the way up to about 55% sugar within that sugary water. Above about 55% sugar, they don't seem to have a preference. There's probably a really interesting reason for this, and I'm going to save that for another time, so you have to come back another year and find out more about that. So what's all this got to do with the strawberries, which I was showing you earlier? Well, when we were doing these, when Emily was doing this experiment to have a look at what sugar concentration the bees preferred, she was using 15 cubic millimeters, which is a drop about, let's say about hemisphere about 3 millimeters wide on these discs. That is an awful lot of nectar. And if you remember, I said my strawberries only produce about half to 1 cubic millimeter of nectar. So I wanted to know, do bees actually still care at these teeny tiny volumes which I'm measuring in strawberries? Because if they don't, there is no point at all in trying to improve the nectar concentration within the strawberries to try to get my bees, or try to get insects to go and visit those strawberries and learn that they have a better one. If they don't care, it's not worth doing. So I wanted to see whether I could repeat this experiment with just 1 cubic millimeter of sugary water. And it turns out that doing that is really difficult, because a bee can drink that in about 2.5 seconds. Bees are also quite fickle creatures. They get bored quite easily. Now, it takes me about half a minute, or when I first started out, it took me about half a minute to reset this experiment, to put another 1 cubic millimeter of sugar water onto the card. And the bee just got bored and flew off. So I needed to work out a way of doing it faster. It took about 6 months on and off to work out how to do it. And in the end, I settled upon this absolutely hellish contraption, which is may best be described as a bodge job. But the thing is, it works. What it is 2 pipettes, so scientific dispensers, which will dispense really accurate volumes. And I've gaffer taped them to a bit of floorboards. And they are spaced just the right amount apart that I can put a card underneath them. You can see it at the bottom. And then there's 2 triggers on them. And if I press those triggers, they squirt out exactly the right volume. And with a bit of practice, I could get this down to about every 5 seconds. And that is fast enough to keep the bee interested. So what I did was I put a card under, squirted the sugar on, and put the card into a flight arena. So I've got one here. This is a sort of green. It's a box with a green floor, and we put a lid on it, and the bee's coming through that hole at the end there. And I could put one card into the flight arena, every 5 or 10 seconds or so. And the bee was interested enough in that. And then I could also record which color she went to. Was it the yellow one for the more sugary solution, or was it the white one for the less sugary solution? It was a bit of a scrabble to do it, I will freely admit. The floor ended up covered in cards as I was discarding them. But I was able to do the experiment, and I was able to show that, yes, bees do still care about a sugar difference, even at 1 millimeter cube volumes. And that's really cool, because it shows that the experiments that we've been doing here do actually have a real world application. And that was something which is really, really important to me when I'm doing my PhD. I want to try to do something which will have a real world use and possibly even change the world, make it a slightly better place for my daughter. So I hope that's explained a bit about what it is that I do, and I hope you've enjoyed seeing a bit about behind the scenes in both my Polytunnel and the Bee Lab here in Cambridge. If you'd like to find out more, then have a look at either the Department of Plant Science's website, or if you go online and search for a video called Improving Flowers to Help Feed the World. That's a video which myself and a friend put together a couple of years ago, which explains our research in a bit more detail. Thanks ever so much for listening. Do take care, and if you have any questions, then just drop me a note. I'm Hamish Symington on Twitter, do drop me a note there, or like I say, go and have a look at the Plant Sciences website. Take care. Thanks for coming.
Brilliant. Well, thank you very much, Hamish. As you heard there, Hamish Symington hopefully will be on hand now to answer some of your questions. Here he is. Brilliant. Okay.
Hello there.
Welcome in real time. Fascinating film, Hamish. And the first question came is, do you ever get stung by the bees when you're getting in and out of the arena?
Lots of people ask this one. I like this question. I have been stung by bumblebees twice in the 4 years that I've been working. Both times were when I really annoyed the bee, so it was entirely my fault and not the bee's fault. But most of the time, I just I can keep my hands in there and they fly around perfectly oblivious. I get stung a lot more by my honeybees, which I have at home.
Okay, so there's a couple of questions here about whether bees can distinguish all colours, and I guess also that would be following up partly by the fact that you were using yellow and white. Would that affect maybe the choice as well as the sugar concentration?
So that's a great question. Let's start out with the distinguishing colours question. Like us, bees have 3 colour receptors in their eyes, but we've got red, green, and blue, and they have ultraviolet, green, and blue. So they're missing a red receptor. It's not they can't see red, because the tail end of the green does detect a little bit of red, but to them, red is what we call achromatic. So it's a really hard concept to explain, but the best way to say it is it looks a bit like grey. Or something like that. So they can't see red very well, but they can see an ultraviolet. And some flowers have patternings which reflect ultraviolet light, and the bees can see those, which we can't. So for example, a dandelion has a nice big ultraviolet absorbing center, and around the edge that reflects ultraviolet. So in bee vision, if you like, there's this big bullseye of flower. So that's the distinguishing colors question. So you asked whether the choice of yellow and white would affect the experiment. That is something which we as researchers need to think about, and that's why for half of the experiment I did it with the high concentration of sugar on the yellow ones, and for half of the experiment I did it with the high concentration on the white ones. And then that will mitigate for any effect that you can possibly get there. But excellent question.
Okay, there's another one here asking about, you mentioned pollen and the importance that you're counting pollen. And there's a question here asking about how do you assess the importance of pollen to the bees relative to nectar?
So bees need both. Pollen is protein, which the bees need to make more bee, in the same way that we need protein to make more human. Nectar is energy, it's carbohydrate, which powers their muscles. So the question is, what's the benefit of having flowers with higher sugar concentration versus just having more flowers as well? Both would be great. If the plant can do it, from the bee's point of view, we could have more flowers to provide more food for the bees. yeah, if we can increase the amount of nectar as well, there's all sorts of different ways that we can tweak these plants to make them better for bees. And it may be that having more flowers would actually make the crops for us less productive. It might make more courgettes or what have you, but smaller ones. So we need to balance everything out there. There's so many different variables involved in this sort of thing.
Okay, thank you. And there was a question about when you dye the pollen, what are you using? What sort of stain are you using? And could you do that if you had a microscope at home?
So the stain I'm using it uses quite a lot of different components. It stains viable pollen deep red and non-viable pollen, so pollen which isn't fertile, it'll stain that blue. So I use 3 different stains. There's an acid, there's lots of concentrated alcohol, 95% alcohol, which is yeah, very, very strong indeed. So these aren't things which you can get at home, but you can just have a look at pollen. If you've got a microscope which uses slides at home, then you can take pollen and have a look at it. Some good ones to have a look at there are mallow flowers. If you can find those little purple ones, they grow scrubbily all around roads and things like that. If you have a look at lily, you'll get some really nice big pollen there. Don't use forget-me-not, because the pollen for that is the smallest pollen that you can get. It's really, really tiny, so you wouldn't be able to see it.
Okay, and then I think a final question before we move on to the next presentation. Can you explain why bees don't care about sugar concentrations above a certain amount?
Oh, that's a really interesting question. I mean, can I have the next hour? Is that
No, we need to move on.
So we're not entirely sure, but it's probably going to be something to do with the rate that they can get energy back to the nest. So it's to do with the drinking rate and the rate that they vomit it out when they get back in the nest. And it's going to be something to do with maximizing the rate of energy transfer back to the nest. But we're not entirely sure. It's a really difficult question to get the answer to for that one. But we are working on it. So watch this space. Maybe come back in a year or two and I'll have a better answer for you then.
Well, thank you very much, Hamish. Hamish, I should say, is doing a PhD within the department, funded through the BBSRC. And he'll be on hand and he'll look at some of the questions that are coming in. And I hope we'll carry on answering some of those questions to you individually by typing answers. But I think we should move on to the next film. And this is a little bit about, well, I'll leave you to see what's coming up. It's somebody being rather cruel to plants. And why would they want to do that? And what sort of pest is attacking these plants, I think would be the question. And there are some audience polls for you to vote during this particular film. So thank you. And thanks to Hamish.
Look at these mysterious holes. What could have happened here? What is this? It looks like the leaf of a plant, but why is it full of holes? This video shows a lot of plants in the greenhouse, and many of them also have the same types of holes in. Whatever caused them must be very determined. What do you think could have made those holes? It could have been an insect or a disease.
But it turns out there's a strange animal on the loose, collecting small circles out of the leaves. It's a scientist. The process of collecting these leaf discs requires highly scientific equipment, a hole punch and some tweezers. There are hundreds and hundreds of these plants in the greenhouse. Does anyone know what type of plant this is? It's a maize plant, which is the plant that produces corn.
We had to collect lots of circular discs out of our maize leaves, so we needed many different scientists to take circles out of the leaves, working late into the evening. Even with our team of 6 scientists, it took 2 weeks to collect all the discs we wanted. We eventually harvested about 20,000 leaf discs. Each disc like this had to be carefully collected for our research. Why did we need to collect our samples as tiny discs? Transporting these discs back to the lab is a lot easier than bringing 800 whole plants.
Each of these plants is a different type of maize, grown from a huge variety of different seeds. We grew them in a luxury air-conditioned greenhouse so that they would receive lots of light to help them grow quickly, and so that they would have enough warmth without becoming overheated. This allowed us to grow healthy plants that were ready for sampling as soon as possible. But what did we want these leaf discs for anyway? Well, once we had collected the leaf discs, we gave them an unusual treatment. We exposed them to a stressful situation, high light and cold temperatures. When plants are stressed, this means they're in conditions that aren't good for them. This can affect the way they grow, and they can end up looking very unhealthy. When we had finished exposing the plants to the stressful situation, we then put the discs into tubes for storage and to enable measurements to see how the stressful treatment affected the leaf discs.
Here you can see about 100 leaf discs. Why are they arranged like this? When we have so many discs to measure in our experiment, we need to keep them organized. Keeping a standard layout like this means we know which sample is where, and makes sure that each experiment is as similar to the others as possible. Once organized in these plastic boxes, we sat the leaf discs on cold plates underneath bright lights. We also took a break for this photo shoot because it was so pretty to look at. But can't have been fun for the leaf discs. It would be like sunbathing on an iceberg. When might a plant experience these conditions in nature? These conditions replicate the most challenging conditions that a maize plant might encounter during a UK winter.
Because we had taken so many leaf discs, there was no way we could measure them all in one day. So after exposing them to the stressful treatment, we needed to store them in the freezer to measure later. Kind of like putting your leftovers into the freezer to eat later. We use liquid nitrogen to freeze our samples. The liquid nitrogen is really cold, much colder than your freezer at home. The liquid nitrogen freezes the samples really quickly so that they are as fresh as possible when we take them out of the freezer later. It also looks really cool. This is a video of the time we froze some grass with our spare liquid nitrogen. And here's a box of frozen samples ready for storage.
After we've exposed the discs to the stressful situation and stored them safely, it's time for us to defrost our samples and make some measurements. We took lots of measurements to properly understand every way that the plants had responded to the stressful treatment. For some measurements, we need to take liquid out of the plants to understand what is happening inside each leaf. We'll show you a couple of examples. In this experiment, chlorophyll has been extracted from the leaf discs, giving us a pretty green colour. Chlorophyll is a special molecule inside the leaf that helps it to carry out photosynthesis, the process that plants use to make food. The darker the green, the more chlorophyll there is in the leaf. The amount of chlorophyll in the leaf tells us how healthy or unhealthy the plants are, which shows how well they dealt with the stressful conditions. Here's a scientist measuring some of the samples in a different way. Why isn't this sample green? When we add different liquids to the leaf discs, different things are extracted from the leaves. This experiment measures how damaged the cell membranes are. We put the leaf sample into some water and measure how much stuff from inside the cells has leaked out into the water using this little white and pink machine. Does water extract chlorophyll? No. If it did, these samples would be green too.
Why do you think we've been doing this? Why do we care if plants are stressed? We are measuring stress because stressed plants don't produce as much food. In this photo, you can see a healthy plant on the left and an unhealthy, stressed plant on the right. In cooler countries like the UK, plants like maize that come from hot countries can be stressed out by cold conditions, especially in high light. We want to understand which maize plants are best able to withstand cold temperatures, so we can help plant breeders and farmers produce better corn in colder climates. This is especially important with a growing global population, because maize is the most produced crop in the world. It is used to feed people as well as to feed animals and produce biofuel. We'll leave you with a fun fact about maize. There are different types of photosynthesis, which is the process plants use to make food for themselves and for us. Most plants use normal photosynthesis, called C3, whereas maize plants carry out a special kind of photosynthesis, called C4. This special photosynthesis is particularly efficient in certain environments. This means that maize plants can convert even more sunlight into food for us to eat, compared to other crops, such as wheat or rice.
Well, that was really fascinating. Thank you very much. I'd like to introduce you now to Angela Burnett, Dr. Angela Burnett, who's a postdoctoral research associate in the Environmental Plant Physiology Group. And she's also a David Mackay Research Associate at Darwin College and in association with Cambridge Zero. And also Elizabeth Torley, who's about to start her third year studying natural sciences here at the university. So we've got a time for some questions. And if you'd like to pop them in the panel in the panel there, I can start off. We haven't got anything directly relevant at the moment, but why is it that maize suffers so much in the UK?
Well, that's an interesting question, Howard. The problem is that maize originated in tropical climates. So places like South America tend to grow a lot of maize where it's much warmer, and unfortunately our weather conditions here are not quite what maize prefers, and that can make it difficult for maize, especially in the late winter and the early spring.
Okay, thank you. Well, we've got a tricky one here that's just come in, whether your work could help choose plants for future missions to planets like the Moon or Mars. I think they must be thinking of that film where they grew potatoes. What's your answer to that one?
Well, that's not what I'm working on directly, but I think that it could be relevant, because when we're looking at how plants respond to different environmental conditions, of course, that's relevant when we're looking at conditions both on Earth and on other planets too. Now, that would be quite different conditions that we'd need plants to face on other planets, but temperature stress, whether that be warm temperatures or very cold temperatures, would certainly be something that would need to be considered for those missions.
I always ask my students whether they've been taken to a maize maze by their parents. So it always fascinates me that wheat is normally being harvested round about July and August, and yet maize is still growing at its most. So what's that difference, do you think, due to?
Well, I guess when plants are planted in different seasons, then they will respond differently to different conditions. So you're right that we'll harvest different crops at different times. And I'm just seeing some more questions coming in actually about this similar topic. The date on which we plant plants will affect the day on which they're harvested. So wheat, for example, in the UK will sow at 2 different times in the year and harvest at different times. Crops from different latitudes have different preferences in terms of how much sunlight they want and the angle of the sun, and that will stimulate their production as well. So how long the days are will determine when they come into flower and when they produce their seeds and grains.
And so following up, we've got a question here about the cold and bright conditions. So what is it about this cold that causes the problems for the plants under the high light, particularly?
Well, it can be quite challenging for the process of photosynthesis, which is the process that plants use to produce food. So when it's high light, plants are trying to do photosynthesis to make the most of all the sunshine. A bit like people in the summer heading out, going sunbathing, going to the beach when they can. But imagine if you were going out to the beach for sunbathing, but it was actually really cold. You'd be a bit confused. On the one hand, you want to be out there in the sun, but on the other hand, it's cold, feeling a bit chilly, you're not feeling very energetic. And so it can be challenging for the machinery inside the leaves that does photosynthesis to know how to handle both the bright light and the cold conditions at the same time.
Thank you. I always, when I talk to the students about this, I always remind people to look at maize, a field of maize in May when it's really yellow and it's lacking chlorophyll. There's a question here about why some of the samples look greener than others. So what's that effect of that highlight or those low temperatures on that chlorophyll content?
Yeah, so often when plants are stressed, their chlorophyll content can decrease because due to the stress that's being caused. So that can make the plants go a bit more yellow, and especially in cold temperature, plants can become a bit more yellow as well. And we did notice actually some of the varieties we were growing would naturally be a bit more yellow than other varieties. And then also we see that the cold temperature makes all the varieties go a bit more yellow too.
And then just one final question, because I realise with time's ticking on a bit, but you told us that these maize plants have got this C4 pathway, and if that's more efficient than C3, the question here is, why aren't all plants C4?
So you'll notice we said a little caveat in that fun fact that it's only beneficial in certain conditions, which specifically is in hotter climates and when water is more limited. And as we said before, maize originated from these types of climates, which is why it has it. And not all plants have it because it comes at a cost as well. They have to set up this special machinery, this special system, and that costs a lot of resources and energy. So other plants that might have evolved in the UK or in Russia, somewhere cold, wouldn't have wanted to spend all of their resources on this system because it's not useful to them.
That's brilliant, Elizabeth. Thank you very much for that really clear explanation. Angela, you too, thank you for all your efforts and all those fascinating insights. And I'm sorry if I inferred that you were pests in making those holes in those leaves. Anyway, let's move on finally to our final presentation. Thank you both. Move on to our final presentation, and this is a live presentation I won't tell you where they are or what they're up to because they're going to explain that to us for themselves. But it's got something to do with, this one has got something to do with astronauts and vitamins.
Well, thank you very much for the introduction, Howard. My name is Payam Mershahi. I'm a senior research associate and the manager of the facility that we are standing in. This is the Algal Innovation Center. And with me here today is Lorraine Archer, Dr. Lorraine Archer, the research coordinator here, and Alan Harrison, who's a PhD student in the group. You'll be hearing from Lorraine and Alan a little bit later on, but I'm going to just tell you about where we are located. The Algal Innovation Center is located in the Botanic Garden of the University of Cambridge, and you can find us when you enter from the Station Road entrance. We're just on the right-hand side before the cafe, and you can see on the map here where we are located. So if you get to the cafe, you've gone too far. Now, I will tell you a little bit about the type of work that we do here, but in the spirit of Through the Keyhole, I wanted to pose a question of how you think the Algal Innovation Center is helping this astronaut wandering aimlessly through a wheat field, individuals who are in India, for example, on strictly vegetarian or vegan diets, and this market that one of our colleagues in Uganda took a picture of. This is actually the back of the market where the fruit and vegetable waste is disposed of. And also, I want to ask how you think the Algal Innovation Center is connected to or is helping towards developing these beautiful products here, the juicy burgers or a vegan mayonnaise or protein bars shown in the picture. I guess the clue is in the fact that we work in the Algal Innovation Center. The algae is the clue, and the company that makes that juicy burger is based in the US. They're called Triton, and their motto is Algae Innovations. So these are all algae-derived food, and we address concerns and challenges that face that astronaut, individuals on vegetarian, vegan diets, and market-based in Uganda or other countries for that matter. At the University of Cambridge, we have a history of working on algae, and there has been numerous developments that have pushed the field forward. And we've had researchers working on uses of algae towards energy, be it biofuels or photovoltaics or biogas generation, to food, feed, pharmaceuticals, using algae for waste remediation, fertilizers, and soil improvers, and we do a lot of global outreach and education programs, so that's why we are here today. The research on algae takes place actually at many sites around the university. In our case, we are located at the Department of Plant Sciences, that historically the research has been taking place there. The Department of Plant Sciences is located at Downing site, and it's been there since 1904. Now, over the decades of work that has gone into algal research allowed us to attract funds to build this facility, the Algal Innovation Center that's shown in the picture here. A photo at the top is in the spring. It's beautiful out in the Botanic Garden, and you can see the Algal Innovation Center, AIC, at its best. Essentially, this is a custom-built facility where we can attract industries or companies and academics to come and test their ideas, test their technologies, advance them, and identify algal-based solutions to the challenges that we face as a society. And at this point, I'm going to pass the mic over to Dr. Lorraine Archer. Thank you, Lorraine.
Hello, welcome to the Algal Innovation Centre. So Hayam has been talking about some of the motivators for the research, some of the research that we carry out here. And that can kind of, a good focal point for that is if we look at the Sustainable Development Goals. And there's a number of those goals, 7 of the 17, that microalgal research can contribute towards actioning. And that's zero hunger, good health, clean water, sustainable cities, responsible production, climate action, and life below water. And one of the ways that we can address many of these is bioremediation of nutrients. So another issue that we can address growing microalgae is the lack of omega-3 oils that are currently in our food systems. So omega-3 oils are necessary in aquaculture, pet food, and in human nutrition. And there's a dwindling supply of available omega-3 oils currently, and that's largely due to a dwindling fish supply due to overfishing and changes in fish population due to climate change. So where do foods get their omega-3 fatty acids? And I'm going to hand you over to Ellen.
Right, so as we've alluded to earlier, fish get their omega-3s from algae, and algae is a really general term for a lot of incredibly diverse group of organisms. So on that slide you can see the whole range of forms that they can take, and we have microalgae and macroalgae, and our lab focuses mainly on microalgae, but overall there are about 70,000 species of microalgae. And that's quite a conservative estimate, but only 50 are currently used for commercial purposes. So that gives you an idea of the scale of different discoveries that are just waiting to happen. In terms of algae as a food or a feed source, most of the ones that we're familiar with are recognised as safe for human consumption. And if we look at the future foods that have been set out, and they're all listed across there, and I've highlighted chicken and soybeans, things that we're quite familiar with, chlorella and spirulina, two algae you've probably heard of, are quite high in protein, and that makes them a really good potential source of nutrients in the future. But compared to the other organisms, they do even better when we're talking about omega-3s, as Lorraine talked about earlier. They're also can be quite high in other vitamins and minerals, and that's really what we're interested in with our other collaborators that we have with projects in India, and we're growing the algae to have a high vitamin B12 content, which is really important for good human nutrition, and it helps with your nervous system development and your motor skills. So we're generating foodstuffs that have high B12 content, and this is particularly important in space. So Howard has promised this astronaut link throughout the whole talk, and my project is funded by the European Space Agency, and a bit like Matt Damon in The Martian, we're trying to colonize Mars. And so on this long space mission, humans would need an edible source of vitamin B12 and other nutrients, and so it works in this closed loop ecosystem concept, and so all the waste from the crew will be digested by bacteria and then fed into various compartments before it gets to the algae and the plant compartments, and that will go on to feed the astronauts, provide oxygen and water, and importantly vitamin B12. But this circular concept can be used on Earth as well, and we're working with partners in Ghana and Uganda to valorize crop waste by anaerobically digesting that and then feeding that digestate of the crop waste to microalgae. And you can actually see part of that project happening in the back corner over here, and then that will produce useful algal biomass and also clean water, and the algae can go on to feed animals. And so this is how we're working to really address those sustainable development goals. And Lorraine is going to talk again about what we do here in the Algal Innovation Centre.
So here in the Algal Innovation Centre, we bring in various different types of algae and we put them through their paces. So we bring in algae in smaller volumes, like you can see here in the flask, and then scale those up in various different photobioreactors. So you can see in the picture there, we have bags hanging of algae that have 10 litres in each bag, and then we can increase the volume of those bags up to 20 litres. So we have a poll. How much algae do you think is growing in our larger reactors there in the middle picture? Okay, well done. So we can grow in those two reactors. We grow about 100 to 120 to 150 litres in the top, and the same in the bottom. And then we can compare the growth of the two. So what Ellen and Payam have been talking about is algae being scaled up for their high-value metabolites that they increase under various stress conditions, or some of them will increase particular metabolites of interest like omega-3 when they're low in nutrients, or when they'll change their metabolite profile when they're hot or when they're cold. So we will put the algae through the paces here, and we don't have to try very hard because we're in a greenhouse, so we get huge variation in temperature. So what kind of temperature would you think the algae would have reached in here in the summer? Well, well done. We reached ambient temperatures in the AIC of 45 degrees. And then those temperatures would have dropped really quickly to about 15 degrees at night. So that's a big change for our single cell cultures to deal with. But these are some of the challenges that we like to address here in the AIC, because they're the questions that are important in terms of growing algae in all kinds of different hostile environments. So I'll hand you back to Payam.
Hey, great. Well, so thanks to Lorraine and Alan for outlining some of the research that we do here at the AIC and what we see as the future of how the algae can contribute to sustainable food and sustainable diet, is that we can really address these challenges and enter the algae, or try to help companies to address shortcomings in our diet. So companies that are in this sector, currently operating in this sector, include Algae. This is a UK-based company that's come up with different types of chlorella. This is a type of algae that is, you may have seen in the shops. They have different colors, different flavors and aromas, and they've already been working with Unilever to incorporate this into, for example, ice cream you should see in the picture. There's a French company called Tamalga that has been involved in making egg substitutes using microalgae, and they've also developed the first vegan mayo from this. Triton, as I mentioned, is a US company that's using Chlamydomonas. This is an algae that we do work with in the lab and here at the AIC. And amazingly, they've used this microalgae to generate meat substitutes. So, such as so these burgers and the tuna that you can see in those cups, or the hot dog, are all derived from Chlamydomonas, grain hearty. And on to the next slide, sorry. This is where I will say thank you for your attention, for listening, and for taking in all of the information about the microalgae. And our final poll, I'm going to pose here to ask you, well, after listening to all of this information about microalgae, how likely are you to actually try an algae-based food in the future?
Positive.
Okay, so we'll take that as a great result. I want to thank all the funding bodies, as well as all the people who have been involved in working in our group and working with us. These are some of the individuals pretending to be flagellated microalgae. And we'll leave you with the next slide, which is Nematococcus that is swimming around under the microscope. And with that, I'll end, and we're happy to answer your questions. Thank you for listening. Okay. Lorraine and Ellen, you've joined.
Should I stop?
Okay, well, thank you very much, Payam, and for your and Lorraine and Ellen, some fascinating insights there. We have some questions. I hope you can hear us okay, and we'll try to go through. So there's a question here. What do algae taste like?
Yum. I think it's, well, we've all tried microalgae, and actually we're all fans of microalgae and algae in general, macroalgae, so seaweed and so on. Some of our Welsh friends will be familiar with lava bread, for example. I know Howard is a big fan, but maybe you guys want to add what algae tastes like to you.
I think it's quite like a dark green vegetable, and it hasn't been processed too much, so quite like some kale, broccoli sort of flavours.
And what I must add is that actually the products that are coming through the market, especially the Algae chlorella range, they have, well, according to the company, they almost have no taste. So the key about generating those products is that they want to take away some of the flavors that has been putting off the people from trying this food, and but still impart the nutritional values that are so important in incorporating algae into diet.
Yeah, thanks. I think a lot of these algae are known as superfoods, aren't they? and the way they have the very good mineral balance and so on. There's a question here about how you process those algae. Won't you get contamination in the algae if you're using wastewater? How do you make sure that that doesn't get into the human supply chain?
That's a very good question. I can answer that. Essentially, we are not allowed to use algae grown on wasted digestate that we talked about for human consumption. There are very strict guidelines in Europe that means the type of waste that is used to grow algae has to be, for example, from vegetable and fruit sources. There cannot be any other source of waste that is anaerobically digested and then the algae are grown on. Mostly then the algae that we do grow is used as feed, and so that's again something that we are careful about, and there are a whole range of tests that need to be taking place to ensure that the bacterial load or pathogens that can be in our product are not present.
Okay, thank you. I mean, there's a question here was asking. I know you did show us. I think it was it Ellen, or you showed us some algae when you held up a flask. Could you bring one of those flasks up to the camera, because you're much you're now bigger relative to the slideshow that we had earlier. Let's have a look. So these are. Do you know, is that Chlamydomonas?
That's chlorella.
That's chlorella. Okay.
We'll be happy to take you for a little walk through the facility if you really want to see the chlorella at larger volume. So,
well, we can see it in those behind you, and I think time's moving on a little, so I think we can see those bags there in real time. That's fantastic. So there's a question here about has anybody got algae as house plants? Do you keep algae at all?
Intentionally?
Well, I could comment, because we used to have a freshwater fish tank, and I tell you, we grew algae in that tank. I don't know how people keep them so clean, but I don't know of anybody who really. You'd get them growing in sometimes with terrestrial plants in a terrarium, won't you? If you have a very moist environment, you'll often see algae growing over the surface. Sometimes they have a kind of a blue-green color, and that indicates that they're cyanobacteria rather than algae.
We actually hope to collect some samples from puddles around Cambridge and show you that, because algae is everywhere, and we did find some particularly nice samples, but they, if you scrape a puddle, you'll find some algae in there. They're everywhere.
And one point about growing algae. So there was a US company that actually approached us, interested in developing their household spirulina growth facility. So this is just a little kind of jar that you would put in your kitchen, and you would grow your own spirulina. I am a little bit hesitant to be involved with that company, because growing algae at home in a media that will allow other microorganisms to grow in may be a recipe for disaster, because you don't really know what else you're growing along with your beautiful spirulina that is a health food, and people are using in pill form and in every other form.
Okay, and then one final question before we draw this to close. We've overrun a little. There's a question here from Dave McKenzie asking about whether there are any limits to growing algae. And I guess for that one, the question is really, you've got these 100, 200 litre containers behind you. What's the most volume you could potentially grow? And is this viable as a means of growing algae on a mass production scale?
Well, in here, that's the largest volumes that we grow, but there are algal producers around the world that would grow in much larger volumes. There's a lot of life cycle analysis being carried out on different reactors at the moment, and there's definitely challenges with energy input and stuff like that in closed reactors that need to be kind of addressed. But that's kind of where it gets interesting in terms of circular economy and taking flue gases and waste energy and nutrients into the life cycle analysis of algae, because then that's when it really comes into its own.
And
absolutely fascinating.
That Triton, the company based in the US, is currently growing 5,000 liter and their bioreactors, and they've increase that capacity to 100,000 liters by the end of the year, they've said, and they continue to expand that. So there is definitely capacity to increase the volume of algae that's being grown, and there are different technologies that are going to be applied to increase that even more in fermentation mode or so.
Okay, well, that's absolutely fantastic. Thank you so much for those insights, delivered brilliantly in real time. That's Payam Mashami, who's Dr. Lorraine Archer, and Ellen Harrison, who's a PhD student, greeting us there from the Algal Innovation Centre down in the Botanic Gardens. Well, I think we've come to the end. Thank you very much for staying with us. I think there should be one final slide we'd like to see, because on this slide it just reminds me to thank everybody who's been participating in this particular program tonight. We heard from Sebastianese van den Akker and his wonderful team, Sukira Ko, Olaf Krantz, Beth, and Clemens Pellegrin, and Beatrice, and a few others. We've also heard from Hamish Symington, and then we also heard from those holy plants with Angela Burnett and Elizabeth Torley, and then finally we've heard from Payam, Lorraine, and Ellen, giving us these fascinating insights. So through the keyhole, finding out how fundamental research is done, and then seeing what possible applications there may be in the wider world. And that's really what excites us scientists. It's doing the fundamental stuff and then finding ways to apply that for the good of humanity. I'd also like to thank particularly the organisers, the backroom people who've been helping put together these shows. Clearly, it took a huge amount of time for everybody to put together their presentations, but I'd also specifically like to thank Karen Hlaber and Lucia Arce Cubas, who've been doing much of the film editing and sorting out, and then finally, Abigail Youngman, who's actually very quietly removed herself from this list, but has actually been the motivating force behind this entire project. So thank you very much indeed, Abigail. And thank you for staying with us and watching, and I hope you found this an informative and entertaining evening. Thank you and good night.